All right. So in our last class, we took a look at photosynthesis and we took a look at the factors that affect the rate of photosynthesis. And we also went over a few experiments, right? So today we're going to take a look at the structure of the leaf and what are the different parts of the leaf. So give me a second. Let me just share one diagram with you guys. Hold on. So a leaf is basically composed of all of these parts, right? The topmost layer is referred to as your upper epidermis. Okay. Epidermis is basically the outermost layer of the cells. Okay. So in any tissue, in any organ, the outermost layer of the cells is your epidermis. So epidermis, the top one is called the upper epidermis. The bottom one is called the lower epidermis. Okay. Then on top of the upper epidermis, we have a waxy cuticle, a waxy material that covers the surface of the leaf. Now, below that, we have our mesophyll cells. Okay. There are two types of mesophyll cells. We have the palisade mesophyll cell and we have the spongy mesophyll cell. The palisade mesophyll cells are called that because they have a tall sort of, like a column like or a rectangular like shape. Spongy mesophyll cells are usually circular and they have large air spaces between them. Okay, that's why we call it spongy because they have air spaces between them. Then below the spongy mesophyll cell, we have the lower epidermis, right? And in our lower epidermis, we have really special cells which are known as your guard cells. Okay. Guard cells are responsible for opening and closing these pores that we call stomata. So they're in the bottom surface of the leaf. Okay. Generally, not always, but generally. In the bottom surface of the leaf, we have these small openings. Okay. Those openings are referred to as stomata. Those openings are controlled by guard cells. So guard cells will open the openings or they will close it. The purpose of the openings or the purpose of the stomata is allow for exchange of gases so that carbon dioxide can move in and out, oxygen can move in and out, water vapors can move in and out, right? So that is why we need these small openings. Then it's not drawn over here in this diagram, but you also have your vascular bundle. A vascular bundle is composed of what? A vascular bundle is composed of your um xylem vessels and phloem vessels. Okay, I'm going to teach you guys in detail about what xylem vessels and phloem vessels are when we do the topic of transport in plants. But mainly speaking, they have the function of what? Of transporting substances throughout the plant. So this is composed of xylem vessel and also phloem vessel. So we call it a vascular bundle. And this has both xylem in it and phloem in it. Xylem is responsible for transporting water, and phloem is responsible for transporting food, basically, your sucrose. Now, let's take a look at each and every one of these parts one by one. Let's start off with the waxy cuticle. Okay. Why do we need a waxy cuticle? A waxy cuticle creates a waterproof layer. Okay. It creates a waterproof layer, minimizing water loss. Imagine if you have plants that are in very hot climate, right? So they would lose a lot of water by evaporation. So to minimize water loss by evaporation, a lot of plants have a waxy cuticle on the top surface. Then you have your upper epidermis. Upper epidermis layer are thin, these cells are thin and they are transparent, meaning that they don't have any chloroplast. Why are they transparent? Why don't they have any chloroplast? So the light can penetrate through, or it can pass through into the deeper layer of the leaf. Then we have our palisade mesophyll cell. Okay. Now, palisade mesophyll cell are the main area that actually contains chloroplast and where photosynthesis actually occurs. Okay. Okay. One more point, sorry, I forgot to add is that the upper epidermis is thin and transparent, so light can penetrate through, and it also prevents bacteria and fungus from getting in and infecting the plant. Okay, add that as well. Then let's come on to our palisade mesophyll cell. Palisade mesophyll cells are the main site for photosynthesis. They are the main sites for photosynthesis. This is where you find the chloroplast. Okay, this is where the chloroplast are majorly found. They're found in other areas of the leaf as well, but majority of them are found in the palisade mesophyll cell. And these chloroplasts over here, they absorb the oxygen, they absorb the carbon dioxide. Sorry, they don't absorb oxygen, they absorb the carbon dioxide, and their main function is to undergo photosynthesis, right? And you guys will also notice that all the chloroplasts are sort of arranged towards the edge of the cell, right? Again, this is to maximize the amount of sunlight absorbed so that all of the chloroplasts are exposed to the sunlight so that they can absorb as much sunlight as possible, because this is where actually your photosynthesis is going to occur, right? And that's why the cells have this sort of column-like or rectangular shape, so that all the chloroplasts are arranged like in a, they're not overlapping. They're sort of arranged like in a more regular manner so that they can absorb a lot of the sunlight. Then we have spongy mesophyll cell. Spongy mesophyll cell is where our gaseous exchange basically takes place, right? It has the main function of the spongy mesophyll is that it has air spaces. Spongy mesophyll also has chloroplast, not as much as the palisade mesophyll, but they also do have chloroplast. They can also do photosynthesis. But the main site is palisade mesophyll, but spongy mesophyll also does photosynthesis. So these are our air spaces. The air spaces is basically allow for exchange of gases, right? These allow for exchange of gases uh, between the environment and the cells. So basically, remember how I told you we have these pores, right? So for example, the carbon dioxide will enter through the pores. And once it enters, it will first go into the air spaces. Then from the air spaces, it will then diffuse into the cell. Okay. So this is your spongy mesophyll. So in spongy mesophyll, the cells are spherical and they're loosely packed. Why loosely packed? Because obviously they have air spaces between them. Then we have our vascular bundle. Okay. Vascular bundle is basically composed of, like I said, xylem and phloem. The xylem is responsible for transport of water and mineral ions. Phloem is responsible for transport of food, which is basically your sucrose, but can include your amino acids and all of that as well. And this phloem xylem bundle, sorry, vascular bundle, right? This makes the vein of the leaf. So for example, if I show you guys a leaf, 1 second, let me show you guys, hold on. So this is how a leaf looks like, right? So this central part is the midrib, and then you have a lot of veins coming out of the leaf, right? So all of this is the vascular bundle. Okay. The vein, the midrib, all of this is actually the vascular bundle. That's what it is. So when you look at a leaf and you see that rib in the middle and then all the branches coming out of it, that is the vein of the leaf. And that is what? That is the xylem and the phloem vessels, basically, which is your vascular bundle. I will teach you guys more about vascular bundle when we do the topic of transport, which is next after this one. Then, after our vascular bundle, we come on to the lower epidermis. The most important feature in the lower epidermis is our stomata, right? Stomata is controlled by the guard cells. So let's study that. That we're going to study in a bit more detail because that's a very important topic. When you talk about the stomata, so stomata is the opening in the pores inside the leaf that allow the gases to diffuse in and out of the leaf. So what actually happens is this, that during the day, right? During the day, the stomata are mostly open so that they can allow carbon dioxide to diffuse in, so photosynthesis can occur, right? During night, uh, they might be closed, right? During night, they can't, it's not necessary. I'm not telling you guys 100% guarantee, but during night, they might possibly be closed because there's no need for them to be open since they photosynthesis is not occurring. And it will also prevent loss of water. If the stomata are closed, water will not be able to diffuse out because through the stomata, um, it's not just the gases that move out, but water vapors can also move in and out. We call that transpiration. I'll teach you guys more about transpiration in the topic of transport. But we call the evaporation of water from the leaf transpiration. So it will minimize transpiration. Now, how exactly does a stomata work? Stomata, the opening of the stomata is controlled by the opening of the guard cells. That's what I told you guys, right? The guard cells control whether they will open or whether they will close, right? So what are we actually going to see? How are the guard cells going to open and close the stomata? To understand this, let's take a look at the structure of a guard cell. I'm going to draw the diagram first and then I'll explain it. Hold on. Okay, so the way a guard cell works is this, that what actually happens is that when there is daylight, okay, the amount of potassium inside the guard cell increases. Okay. Why does the amount of potassium increase? We don't know. The exact mechanism of how this is happening is still not understood. The scientists are not 100% sure why it actually happens. Okay. But what basically happens is this, that due to some reason, when there is daylight, the amount of potassium inside the guard cell increases. Now, over here, we need to relate this light to the topic of diffusion or osmosis. So what basically is going to happen is that the water potential inside the guard cell decreases. When the water potential decreases, what will happen? The water will move from high concentration. So the surrounding cells, the epidermal cells, okay, they have a higher amount of water. The cells, the water inside the guard cells is lesser because the amount of potassium is increased. So the water moves in from an area of high potential to low potential through the process of osmosis. Now, when the water moves into the guard cell, what happens? The guard cell is going to swell up, right? It will swell up and it will end up becoming firm. It will end up becoming a bit firm. So for that, we use a special term. Okay. That special term is known as turgid. Okay. We say that the plant has become turgid. So what happens that the guard cell swells up and becomes turgid. Now, a guard cell is a very special cell. In some areas, the cell wall is very thick. In some area, the cell wall is very thin. So as a result, it will stretch. It will become more firm, but it's going to, in some areas, it will firm up even more. In some areas, it will firm up less. That's why when a guard cell opens up, it gets this kidney bean shape. Okay. From certain areas, it opens up more, stretches more, and some areas it stretches less. That's because of different amount of cell wall. So this causes the guard cell to open. This causes the guard cells to open the stomata. So this is how your guard cell works, right? And for the closing, it will be the opposite. The amount of potassium will decrease, the water potential will increase, the water will move out of the guard cell, and the stomata is going to close, right? So guard cells are the responsible for opening and closing of the stomata. Making sense so far? Does anybody have any question or any part that's not clear? Haja, Maria, is everything all clear? Any confusion in any part?
No.
Mariam, all clear?
Yeah, it's clear.
Okay. Now let's look at some adaptations that leaves have that allow them to undergo photosynthesis. So plants have developed certain adaptations. Adaptation, remember we studied about them in natural selection and all, that that organisms develop certain features that increase their chance of survival. So like camels have humps that allows them to retain water. Polar bears have that fur that keeps the heat trapped in cold climates. So a lot of these, your walruses, seals, they have a lot of fat, which we call blubber, that helps in again loss of heat. So a lot of the organisms have a lot of special features that allow them to survive or have a better chance of survival. So plants also have certain adaptation. Number one, plants usually have a broad flat shape. Not true for all plants. There are some plants that yet generally tend to have narrower shape. Okay. But generally speaking, they tend to have a broad, wide, flat shape that helps increase the surface area for absorption of carbon dioxide and light energy. Increase synthesis. Then, along with this, the leaves are generally thin, right? If you ever touch a leaf, you guys know how thin these leaves are. So the leaves are thin. Again, why are they thin? So that the carbon dioxide only has to diffuse a short distance. So that it diffuses faster. Then, when we talk about it, the large air spaces, right? The large air spaces allow for easy passage through which carbon dioxide can diffuse. Then there are many stomata on the lower epidermis to allow for gas exchange. And there are more chloroplasts in palisade and spongy mesophyll cells because they receive the most sunlight. Now, one last thing that we have to go over in this chapter is a small experiment about how the mineral ions actually affect the growth of the plant. So remember in the last class I told you what does magnesium deficiency do, what does potassium deficiency do. So now let's just take a look at a small experiment. So for that, let me share a diagram. Hold on. So the experiment is done something like this, where you first thing is main thing is that you always have a control. Okay. Why do we need a control? We need a control so that we can compare our results to something that is basically normal, right? Imagine, okay, remember this. Every single experiments that you guys do, every single experiment, not talking about now, A levels, your grads, your post grads, you guys go into research, you guys go into experimental. So remember this, for any research of yours to ever be a success, there should be a control. There should be a normal that you compare it to. Doesn't have to be a normal, but there should be a control. For example, you are looking at the effects of smoking on blood pressure. So if you take a group of smokers, you have to compare them to non-smokers. Otherwise, how will you get your data, right? If you have invented a new drug and you want to figure out if the new drug is any good, you will compare them to people who get the conventional drug, who get the drug that has already been there for many, many years, right? So you always need a control. So our control is going to be full nutrients, healthy growth. Okay, so our control is going to be that you make a solution that has all the nutrients in it. Okay, then you can prepare a solution without any nitrate ions. Then you can prepare a solution without any magnesium. Then you can prepare a solution without any phosphate. It's not given in the diagram, but it will be the same. You draw a solution without any phosphate. Then you draw a solution without any calcium. Then even you can do a solution with just distilled water. No nutrition, all the nutrients are missing. And then you will look at the results, right? For the results, you guys can measure the weight, dry weight. Okay. Dry weight is if you dry them up and then you measure them, dry weight. Or you can measure the length of the plant, right? How much did the plant grow? So you are going to basically, what are you going to see in magnesium? You already know this, but I'll just share again with you another diagram. With magnesium, you will see your plants are going to have a bit of a yellow color. With nitrate ions, you will see that the plants are going to be small, they're going to be stunted, they're going to have the growth is going to be quite slow, the growth is going to be quite less. With with phosphate, again, you're going to see that the growth is going to be quite less. With potassium, you will see that the leaves will be yellow. With magnesium, also, you will see that the leaves will be yellow. Then you can also either measure the length of the shoot, or you can actually go ahead and measure the dry weight. All right. So this is how you can go ahead and perform your experiment. Does anybody have any question, any part that's not clear, any part that did not make sense?
No.
Mayam, is everything all clear?
Yes, so far.
Okay, perfect. So this is all we had to do in the topic of our photosynthesis and structure of leaf, right? So now we have to go ahead and in our next class, we'll take a look at transport. So in which we will go over xylem and phloem. And then we will take, so we in, Haja, I just want to confirm with you, right? We have, you and I have done biological molecules, enzymes. We have done plant nutrition now. We have done digestion in humans. We have done your cardiovascular system, diseases and immunity, respiration, excretion, nervous system, reproduction in humans, inheritance, variance and selection, organisms and the environment, human influences, and biotechnology. Right, Hajra? I think these all we have covered so far.
Yeah.
Okay, so we have done plant nutrition. So we are left with our coordination in plants and reproduction in plants and transport in plants. And then we have small topics left, like your movement in and out of cells, classification, and all of that. All right. Okay, guys, we'll go ahead and stop it over here for today, and we will continue in the next class. All right. Okay, thank you so much, guys. I'll see you in the next class. Have a good day.